The UCSC Cell Browser is a widely used online browser helpful for visualizing a chromatin accessibility atlas in addition to a transcriptome atlas. The Retina landing page contains data collections for human and mouse retina cell atlases. If we open the Human Retina Cell Atlas, we can see it contains a collection of single-cell RNA, single nuclear RNA, and single nuclear ATAC data, as well as the combined datasets of both modalities. For this tutorial, let's look at the combined dataset for bipolar cells. This will open up the embedding for this dataset. You can color and label the embedding by categorical variables on the left, which is currently colored by cell type. UCSC allows you to mismatch the color and label of the embedding too, like coloring by group 2 and labeling by cell type. You can also export the legend on the right, which will download a TSV file which contains the frequency and cell count of each category you have colored. You can view gene expression on the embedding by switching to the Gene tab on the left and searching for a gene. For example, MACE2 is highly expressed in one of the BC clusters. You can also export this visualization at any time by going to File and downloading as either a PNG or SVG. One of the unique features of the UCSC Cell Browser is the ability to generate violin plots by cluster for the expression of the gene. This is done by clicking Gene Expression at the top and clicking Show Violins after searching for your gene of interest. Unfortunately, there is no function to export these violin plots. Once you're done, you can close out of this window. Since it has both RNA and ATAC-seq data, another unique functionality of the UCSC Cell Browser is to view the RNA and ATAC embeddings side by side. To do this, simply go to the View tab at the top, selecting Split Screen, and it'll duplicate your embedding. Next, select one of the embeddings and search for your gene of interest. And then select the other embedding and search for a gene of interest with the ATAC - prefix. Now you can view both the RNA and ATAC modality side by side of a gene. We can also visualize the chromatin accessibility profiles using the UCSC Genome Browser tracks. For example, you can examine ATAC peaks around MACE2 for bipolar cell types. From here, go to File, Open Dataset, and go to the Track Hub link. Here, you can search for your gene of interest, MACE2 in our case. And you can show tracks specifically for bipolar types while hiding others. After zooming out, you can visualize ATAC peaks around MACE2 in bipolar cells. This function is useful for exploring chromatin accessibility profiles in genomic regions.